Hey biology, so today we're going to go ahead and talk about 11.3. So we're going to explore Mendelian genetics. Okay, so genes are more complicated than what Mendel thought. He went ahead and researched the pea plant. Pea plants are very simple, they're not very complex. And luckily, because he chose the pea plant, he did find or he was able to find the information that helped us understand genes. However, it's more complicated, like we said, than expected. So for instance, the environment can influence the expression of genes. So nature versus nurture. Genes provide the plan for the development, but how the plan unfolds also depends on environmental conditions. Some traits have more than just two allele choices. So if you remember, we called that if there's more than two, multiple allele traits. So an example of that is blood type. We have allele choices A, B, or O. Some traits are determined by more than one gene. So remember, the prefix poly means multiple, so we have multiple gene trait or polygenic trait. An example of that is human height, intelligence, skin color, and eye color. Traits determined by more than one gene have many in between phenotypes. So there aren't just smart people and dumb people, there is a whole range of intelligence. So there's also different kinds of dominance. We have complete dominance, and that's when basically dominant always masks the recessive. We have incomplete dominance, and we also have codominance. So incomplete dominance, dominant allele masks the recessive. The pattern is recessive allele returns in the, hopefully you'll say, F2 generation in a 3-to-1 ratio. So in incomplete dominance, we don't see the expected 3-to-1 ratio, where we actually see the heterozygote organisms with the dominant and recessive allele both showing, or like a blend between the two. So incomplete dominance, one's not completely dominant over the other. It's blended. Codominance, both traits are expressed at the same time. So we would say no blending in the heterozygotes, but we see both of them. So a roan horse has both red hair and white hair side by side. Okay guys, so now we're going to talk about when we see both multiple allele traits, codominant traits, and complete dominant traits. An example of that is going to be in blood typing. So multiple alleles, we know that we see an A, B, or O blood type. And that's an example of our multiple allele trait. Well, also in that, we see codominance here. So when you see the AB individuals, you will see that they will express both the A, B blood types. So what does it mean to have a blood type? The blood types, basically, they're based off of glycoproteins found on the outside of your cell. The glycoproteins help the cell recognize foreign invaders inside of our bodies. And so if you are given blood from someone else that doesn't have a similar blood type or a blood type that you can accept, your body will reject that cell and break it down. Okay, so that's basically what our blood types mean. So in our blood types, we're given an allele from mom, or for instance from an AB blood type, we could be given an allele from mom that might be the A allele, and an allele from dad that might be the B allele. Now, codominance tells us that both A and B alleles will be shown. And so around our cells will be both the AB glycoproteins. If you are an A individual, you'll get an allele from mom that could be the A, and you could get an allele from dad that would be the O. Now, A is actually dominant over O. A will be the only thing that shows up, and O glycoproteins actually don't show anything. So what we're going to see is the A just around our cells. In a B individual, B is also dominant over O. So in a cell, let's say mom donates the B allele and dad donates the O allele, we're going to go ahead and see only the B allele show up. And again, that's dominant over the O. So that shows you an example not only of codominance with the AB, but it shows you an example of complete dominance, where something's completely dominant over the other allele. So what would an O individual look like? Okay, so O individuals, if we go ahead and draw an example at the top, means that you would have gotten an O allele from mom and an O allele from dad. They wouldn't have anything or any glycoproteins around their cells. Alright, so why don't we go ahead and do a cross between two different parents of varying blood types, and what would that Punnett square look like? Okay, so let's say that we have a mom that has the blood type AB. Now we represent blood types by using a dominant and recessive I, because we also know that the A and B are dominant to O. So a mom that has an AB blood type, we would represent with a dominant I and then a superscript A. And then if she has a B allele as well, dominant I, superscript B. A dad, let's say, has the B blood type, but he has a B allele and he also has the O allele. So we're going to represent that O allele with a recessive I. So again, it just shows you that we have codominance, the A, B both show up, and we have complete dominance. The B is just going to show up in those individuals. So when we do the cross, let's go ahead and put the mom on top and dad to the left. Okay, so as we've done this before, you know that we want to go ahead and we only put, let's say if we're talking about mom's alleles will only go in the first column, or mom's first allele, the A allele will only go in the first column, the second allele will only go, the B allele will only go in the last column. Now A always comes before B. You never hear BA blood type, you always hear AB blood type. And so I'm going to put the A allele all the way to the left and the B allele all the way to the right. Okay, so let's go ahead then and fill in dad's alleles. Now down at the bottom, you want to put in the recessive I second, always. So in that last one, we're going to have to erase that, and we're going to have to make sure the IB comes before the recessive I. Alright, well now let's go ahead and talk about our phenotypic and genotypic ratios. So the first one we see is we see the AB phenotype, or so that in the genotype would look like dominant IA, dominant IB. That would give us an AB phenotype, and we see that at 25%. Okay, so the next one we have the B genotype. So we see a dominant IB, dominant IB, and that would give us the B phenotype. Now with this one, we only see one of those, 25%. Okay, what about the A phenotype? So we see dominant A, I, and so we see only one of those, that would be an A phenotype, and we see that at 25%. And then last but not least, we see another B phenotype. So we're going to have to change our percentage here. So we have the dominant I and B, since the dominant masks the recessive O. So that would be a B individual. And so we're going to have to go ahead and change that 25% of our individuals.
We'll have that B phenotype.
Okay guys, go ahead and do these problems in your notebooks and then open up to the Rudolph genetics. Now, when we were talking about Rudolph and Rhonda, we found out their genotype, and the genotypes were as follows. So Rudolph is first, Rhonda is second. Now, instead of having to do a dihybrid cross, we can actually use math to figure this out, and you have to have an idea of what the Punnett square might be before each trait. So if we were to cross these two individuals, the only outcome we would get would be the heterozygote and the homozygous recessive. And we would see that in a 2 to 4 ratio. So 2 out of 4 of them would have that genotype. Then if we look at the next two traits, so for flightlessness, and we did the Punnett square for that, we would see the same thing. We would have a heterozygote, or we would have the homozygous recessive. And so we want to go ahead and fill those out and put the probabilities right next to them. So 2 of 4 would be heterozygotes, 2 of 4 would be homozygous recessive. Okay, so then we can figure out the probabilities of those certain genotypes. Of the heterozygote for both traits, we would see, and then we have heterozygote for nose color and then homozygous recessive for flight color. And we would fill in all of those across. So homozygous recessive for this one, messed up there, and then we have homozygous recessive for both of them. It looks like I just didn't do that part. There we go. Okay, so now what we do is we want to figure out the probabilities of seeing that genotype. 4 times 4 would be 16. Okay, and so we're just going to multiply our fractions. 4 times 4 is 16, 4 times 4 is 16. And remember, if you do a dihybrid cross, you have 16 probabilities, which makes sense. So then we go ahead and we multiply the top part of the fraction. So 2 times 2 is 4, 2 times 2 is 4, and then 2 times 2 is 4. If we added up all of those 4s, we would get 16. So we found out all of the genotypic probabilities. Now, to find out the phenotypic probabilities, you would just have to describe what those individuals would look like, and you could go from there. So now what we're going to do is go ahead and try our challenge problem, and you're going to do that via the p-plants. And so you'll want to make sure that you copy down this part, so the p-plants for your challenge problem. So we know that in p-plants, we can have either round seeds or wrinkled seeds. So our round seeds, those are going to be dominant, and we're going to use a dominant R for them. Wrinkled seeds are going to be recessive, so we'll use the lowercase r. Okay, and then we have seed color. Okay, so we're going to have yellow seeds. Those are going to be dominant, and we're going to use a capital Y. And then white seeds, or sorry, green seeds, my bad, are going to be recessive, so lowercase y. Then we have seed color, or coat, so those colors there. We have the gray, the gray coat, and we're going to use capital G for dominant, and white coat, which is recessive, so lowercase g. And then last but not least, we have height. And so height, we're going to use a dominant T or a capital T for the dominant allele, and that stands for tall. And recessive T or short will be the lowercase t. So what I want you to do is go ahead and do a cross between 2 individuals, and you're going to actually follow 4 traits. And so I want you to cross these 2 individuals. So we have a homozygous for round, heterozygous for yellow, homozygous white, and then heterozygous tall. And then our second pair, which is heterozygous for all of those traits.